Today, my guest is Professor Lucia Aronica. She is a PhD and professor at Stanford University, but she's taken sort of a circuitous path to get there. She's originally from Italy, and you'll hear in her lovely accent that she is pure Italian. And then she got her PhD in molecular biology in Austria. She trained at Oxford and then found her way to Stanford specifically to do genetic and epigenetic analysis of the diet fit study. So in this interview, we get into the science. She loves science. You're going to see her passion for the science and the epigenetics, and we talk a lot about what epigenetics are and the research behind them. But we also talk about some very simple concepts like gender stereotypes and how that impacts the diet fits results, and how you structure diets and how you structure studies and how that even is going to impact the results. I think you'll enjoy her passion. If you get a little lost in the science, stick with it because she has some great gems at the end where her personality shines and about how we just should be acting as human beings and as scientists, acting as kids for the love of knowledge and not so much about the wars and the battles, which I think is great for all of us. In your progress, you talked about epigenetics, you talked about food as a biological substance, as medicine, as affecting our genes. So give us sort of the overview of those topics and how you see their importance to health.
Yes, the prefix epi means on the top. So epigenetics, epigenetic marks are molecular switches on the top of our DNA that can turn genes on and off, just like a dimmer switch modulates lights up and down in the room. And this can explain why DNA isn't destiny. Why, for example, you have the same DNA molecule in every single cell of your body, and yet your skin cells look different from your brain cells. So just think of your DNA as hardware, then the epigenome, the epigenetics, is the software that is different in every single cell of your body and turns on and off different genes in different cells so that they look and perform differently. And even animals and people that have the same genetics, but different epigenetics, can look very different from each other. Think of identical twins, but also the caterpillar and the butterfly, queen bees and worker bees. They've all the same DNA, but they can look very different from each other. Even identical twins can develop different predisposition to diseases as they get older. And I think one exciting property of epigenetics is that some epigenetic marks are permanent, as if written with a pen, but some others are written with a pencil, so they are potentially reversible. It's good news that some epigenetic marks are written with a pen, so that, for example, your skin cells don't suddenly turn into brain cells. But it's also good news that some epigenetic marks are written with a pencil because it means that lifestyle can change your epigenetics. It means that lifestyle is biological information. So food is not only calories, but the biological information. Exercise also is not only about burning calories, but it's biological information. Emotions are biological information, and our health is actually a book in progress that we write every day using our genes as the paper, our lifestyle as information, and epigenetics as either the pen or the pencil.
A great description. And I think that's so important because so many people think their genetics is their destiny. And it's clear that that's not the case, that we can change. And the way you said it, food can change our gene expression, exercise can change our gene expression, and even how we feel in our emotions can change our gene expression. Now, that's something we didn't know, even maybe 10 years ago? Like, how new of a study is this?
Yeah, no, it's we the field of epigenetics is has been booming in the last slowly in the last 20 years, and then probably in the past 10 years has been really booming. And I was actually very lucky to start my PhD when the Nobel Prize for RNA interference, which is just a kind of epigenetic information, was awarded in 2006. So I felt thrilled of starting to about starting to work in this field.
Yeah, right place at the right time to kick off this great career. Now, so that interest is what brought you to Stanford to work on the diet fit study. And I'll give a quick summary, and then you can sort of take it from there, I guess. But it was a follow up to the A to Z trial. The A to Z trial showed that the people who were on the Atkins lost more weight than Ornish and Zone and other trials. And then the diet fits, as you mentioned, was the largest study to compare low carb versus low fat in a randomized trial. And at the end, it was considered a negative study, that there was no difference. But I think the details kind of speak a little bit differently. So tell us a little bit about your perspective of the diet fits trial, what it shows and what it doesn't show, and kind of what you're looking at as the next step.
In the primary analysis of the study, which was published in 2018, we didn't find any significant difference in weight loss between the low carb and the low fat group. But to interpret these results, we need to consider an important aspect of the study design that led to some overlap in macronutrients distribution between the low carb and the low fat diet. And this is for two reasons. First, both diets were focused on quality, so whole food, and they both minimized refined grains and carbohydrates and sugar. So they were both lower in carbohydrates compared to a standard American diet. Second, the participants were instructed to strictly limit their carbohydrate or fat intake only during the first 3 months of the study, and then they were allowed to slowly titrate back carbohydrates and fats up to a level that they thought was sustainable for life. The goal here was to make the results actionable for most people and relevant for public health. And in any dietary intervention, there is a trade-off, some trade-off between science and public health. A more science-focused design, such as, for example, a metabolic ward study, is better at addressing the question of whether a diet is effective, whereas a more public health focused design is more effective at addressing the question of whether a diet is sustainable. But I think it's important to stress that these are two very different questions, and that the question of whether a diet is sustainable or not and what constitutes a sustainable diet is a very tricky one. For example, for you guys in Sweden, it might be easier to embrace a low carbohydrate diet than for us in Italy, because we love our pizza, spaghetti, and tortellini. But this doesn't mean that Italian people cannot enjoy a low carbohydrate diet. After all, my family and I have been enjoying a low carb diet now for more than 10 years. And here I have perhaps a piece of advice to share with anyone who wants to reduce their refined carbohydrate intake, whether it's about spaghetti, macaroni and cheese, bread. Um, so it's becoming smart hedonists. So my Italian friends always tell me, Lucia, I prefer to keep eating my spaghetti and have a shorter but happier life. And I always tell them, I also love pleasures. I just use my pleasures and don't let them use me. I'm a smart hedonist. I think that we humans love having the same routine, wearing the same clothes, and having similar foods every day. And we can use this nature to select and stick to those habits that make us happier and healthier in the long run. After all, I think we can all agree that feeling great and having the energy to work, exercise, and take care of our family are life's greatest pleasures.
Great point. I mean, that's what's priceless. And that's why there's not one diet for everybody. And
Yes, no,
when we
absolutely.
promote low carb diets, we promote it as an option for people that I think we need to talk about more as a potential option, but not as the one diet for everybody. And that's part of what bothers me about, some pushing vegan diet as the one diet for the whole world, like with the Eat Lancet report. And that's just bound to fail because there isn't one diet for everybody. No, to rewind for a second though, and to talk about the setup for the Diet Fit study, you said it in a very polite and balanced way. But the way I would say it was, this makes me mad. I mean, this really upsets me, the way that they started at 30 grams per day, ended at 130 grams, and the low fat group was at 210 grams of carbohydrate. So it was 130 versus 210, and they still report it as low fat versus low carb, because then it becomes a misnomer. It's no longer a low carb study. So that's part of what really bothers me about this trial. And I get it. They designed it to be sort of a real world sustainability trial. But if you design it in a way that when you interact with people and you say, look, this is a pretty hard diet to stick with. So I want you to start with 30 carbs, do it just for 3 months, and then you don't have to do it anymore because I know it's hard to do. Versus, look, restricting carbs may seem hard at first, but look at all these examples of these hundreds and hundreds, if not thousands of people who have done it for years. It's something people can stick to if you really stick with it. And the reason why I make a big point about that is because it's the same with a physician. It's the same with a nutritionist. If you sit down with them and they say, wow, this is really hard to stick with, I don't think you're going to be able to do it. That's like a self-fulfilling prophecy. So the same for the study. So I'm sort of just venting here, and I'm probably preaching to the choir with you, but I think that's a problem with the study design. But you're not accepting just on face value that it wasn't that it was a negative trial with no difference in terms of weight loss or in terms of benefit for insulin resistance or genetics, because it sounds like you're taking it to the next step to analyze things even further. So what is on the horizon for you with how you're analyzing the data further to find more nuance and detail?
I'm working on three personalized nutrition projects within the Stanford Diet Fits to look at whether biological differences between people, epigenetics, genetics, and sex differences can affect the response to a low carbohydrate or a low fat diet. For the epigenetic project, I'm focusing on an epigenetic biomarker of type 2 diabetes and asking, is this biomarker written with a pencil? Can we reverse it with a weight loss diet? Is there a difference between the low carb and low fat diet? We have a hint that this might be the case because these epigenetic biomarker has been shown to go up. So the risk of type 2 diabetes goes up when triglycerides go up and HDL goes down. And we see that these two blood lipids are more beneficially affected by a low carb than a low fat diet. I'm also going to work on another epigenetic project and look at whether after weight loss people become epigenetically younger. Epigenetics provide a way of measuring our age biologically. This is called biological age. It is the age of our cells and tissue, and it can be very different from our chronological age, which we typically measure by counting our birthdays. I'm going to work on this project with Professor Karl-Heinz Wagner at the University of Vienna and Professor Steve Horvath, who recently gave a TED talk on the topic of biological age and also a lecture for my online course on diet and gene expression. You can find this lecture on my YouTube channel. I think it's a very exciting and informative lecture. The second project is the project with the genetics. And I'm starting this project with Professor Jose Ordovas at Tufts University. We are going to expand on our primary analysis, in which we looked at only 3 genes, and we couldn't find any effect of genetics on the response to a low carb or a low fat diet.
Yeah, I want to interrupt for a second right there because I think that's such an important point. That there were 3 genes that were tested in the original Diet Fit study. So to say that the nutrition, whether low carb or not, had no relationship to genetics. Like that's just a broad statement. No relationship to genetics, as opposed to saying no relationship to the 3 genes that we studied. Two totally different statements, right?
But I get mad every time I read the health news. I'm trying to teach my students also how to read the health news.
That's important. That's probably the most important lesson you can teach them. Yeah. Sort of your philosophy on science and scientists, and we need to be more like children and be more curious. Tell us a little bit about your insight there and your philosophy there, because I think it's so interesting.
Yes, I think I might be an idealist, but I think that humans have an inborn love for knowledge, and we can use this knowledge to improve our lives. So we have all been children, and we have all loved to ask questions to our parents. And I think that we can awaken our inner child every time we read the health news, or even every time uh, we challenge our beliefs, we review our habits and thoughts, and just asking questions, I think can help us improve the way we do nutrition, we communicate with people, and my message, I think, final closing remark I would like to make is that making questions can help us move from polarization to personalization in nutrition, and make love and not war with food. Polarization is what we see in the health news and social media. It's a natural human instinct. It's our need of belonging to a tribe, a group of people with similar beliefs that make us feel safe and supported. But I think that polarization comes to with a high cost for public health, and goes against the essence of science, which is more about asking questions than thinking of having the answers. And so I think that by asking questions, we can bring everyone into the conversation and change science and society.
Yeah, it's a wonderful, wonderful way to end it on that note. Very cheerful, optimistic note of what science can be and how we can all learn. And with scientists and individuals, we can all learn by asking questions and not being so dogmatic, but actually searching for answers. I think that's wonderful.